So I am thrilled to kick off this session as your moderator. The focus today is going to be on understanding the data that is collected through our MiCorps Cooperative Lakes Monitoring Program, and who better to present that information to you and answer your question than our own Erick Elgin. He is our new Lakes Program Manager for MiCorps, and he's got a long career and history of working with lake data and understanding lake ecology. So with that, I'm going to hand it over to him. Welcome, Erick.
Thanks, Joe. And no better. Wow, that's nice. Thank you for that. Can you see my screen, the correct screen? Excellent. Yeah, thank you, Joe. As you saw in the first part, my name is Erick Elgin, the new lake manager for the CLMP, Lake Program Manager. I made this, somebody asked about, are we splitting Paul's Steen into two? What Paul and I worked on this together, so that's why he's a co-author on this. Yeah, so this is an interesting, I love talking about all this. This is going to be an interesting talk. There's a lot of parameters we're going to talk about because we collect a lot of parameters here in the CLMP. And we collect all of this data through time. It's wonderful. We see the value in it. But then it comes to, first of all, we get the numbers. We have to understand, well, what are we looking at when we see these numbers and when we see the reports? And then also, what does this mean? And that is an incredibly difficult task that not one presentation can show everything, but I hope to give some enlightenment to the data you collect. And then also maybe when you get your reports, you can understand them a bit more. So there we go. The condition of a lake is influenced by many factors. When we think about why is a lake the way it is, we look internally into that lake and we look at the physical, chemical, and biological components, and then how they interact with one another to give us what we see in our lake or what is happening in that water body. And so just as an example, let's say you are in a deep lake that is thermally stratified. So you have warm water on top of cold water in the summertime. And let's say it's August and it's a eutrophic lake. A eutrophic lake is one with lots of nutrients and lots of algae production. Well, that can cause phosphorus in the sediments to release from iron and go into the water column. So because of the physics of the lake, we now have a chemical reaction occurring. Well, once the phosphorus is released into the water column, it might be able to get used if that reaches the sunlight area. And so when the lake stratification breaks up, like it's done in the last month or so, depending on where you are in Michigan, those nutrients might go up into the water column when there's enough sunlight, and then you get an algal bloom that can occur. And so now you can see that's the quickest explanation of how we have a dynamic system where there's a lot of interplay between the physics, the chemistry, and the biology. Now, a lake is not separate from the world around us. It's not disconnected. And so we also have external inputs and processes taking place in the watershed or the land that drains into the lake around it. And so, for example, shoreline development can greatly influence these factors, and you can get a certain condition of the lake because of that. Or let's say the land uses heavy agriculture. That releases nutrients and sediments into the waterways, which can impact our lakes. So a lake is not disconnected from the landscape around it. And what's happening in the lake is affected by the outside. One way that we try to understand the condition of our lake and then therefore be able to describe and predict what is happening is we look at the trophic status. And so the trophic status is a description of how productive a lake is. And what does productivity mean is the amount of plant or animal life that a lake can support. So we would say primary productivity is the amount of plant and algae growth within a water body. And that can tell us a tremendous amount. And we use data that we collect here in the CLMP, like transparency, total phosphorus, and chlorophyll, to tell us what trophic state we are in. And you probably seen these terms. Bob in the previous presentation brought up what does hypereutrophic mean? Well, that is a certain trophic state. And so here are the three main ones. Trophic status is actually a continuum, and I'll be talking about it a little bit later in this presentation. But just quickly for the introduction is that we have low nutrient lakes that have typically clearer water because they don't have as much algal and plant growth. And then this translates into our fisheries and other aquatic animal communities that we have, and we call those oligotrophic lakes. We can think of these as our deep cold water lakes. Joe Noner mentioned Cisco lakes, for example. Cisco lakes tend to be oligotrophic. As we become more productive, the water clarity starts to decline a bit. We have more plant and animal growth. They're typically a bit shallower, and so that's mesotrophic. And then on the far end, we have lakes that have more nutrients, more algal and more plant biomass, and that can translate to many different factors within the lake. Now, lakes fall within all three of these naturally. So some lakes are oligotrophic, some lakes are eutrophic, some lakes fall in between. And you could see from Tamara's presentation that here in the CLMP, we have lakes that fall into all three categories. And just to note, there is another category out here that we call hypereutrophic, and that's where it is usually those are degraded by humans. We don't typically have a natural hypereutrophic lake, and that has a lot of nutrients and a lot of algae. But again, more on trophic status later as I blend in kind of what we do. So how do we get to know more? How do we understand the condition of the trophic status of our lakes? We take a pulse of our lakes. We can take a pulse by looking at the different parameters that we collect here at the CLMP. We collect a wide variety from water clarity. That's our Secchi Disk transparency. Joe said it right, as a 5-year-old or as a young boy, putting down this weird disc in the water, and that can tell you a whole lot about a water body. We do phosphorus, chlorophyll A, which is a proxy for algae, dissolved oxygen and temperature, which is really neat, and I look forward to talking more about it, aquatic plants, aquatic invasive plants, and then also shorelines. All of these together, separately they tell us something, and together they can complete a bigger picture of what is happening with your water body. So we collect a lot of data in the CLMP. These are all the lakes that have ever collected data for the CLMP since the 1970s. I believe it's 1974. And all of this is publicly available. You can go onto our website, click on the data exchange, and then you can get this information here. So all publicly available. And if you are a MiCorps or a CLMP volunteer, you will also get a report or have access to a report. And this report is a summary of the data that you collected for that year, but then also comparing it to years in the past, because the CLMP is a long-term citizen science program. So we want to be collecting data long-term because, as Joe Noner said, that gives us the best glimpse at what is happening in that water body. So the reports are made from this data. So you can access the data by looking at it in this format in our data exchange. You can also download it in Excel, where you'll get the raw data, meaning just the numbers organized in a way that you can start to create graphs on your own or analyze or just take a peek and see what's happening. Everything is derived from that. There are certain parts of the report from in regards to aquatic plants that are derived from other sources, but mostly everything is available in our data exchange that you can download. So we have a high standard for our data because we want it to be useful. And so we do have requirements to make this data as useful as possible for not only the lake, but then also the state. Just as an example, some of the requirements are the number of times you collect. So Secchi Disk readings are important to get across the entire growing season. And so we require at least 8 measurements of Secchi Disk evenly spread out through the summer. If you only have 3, or if you have 8 and they're clustered into May, that doesn't necessarily tell us everything and the data would not be used. And that's the same with other data as well. Without that, you would see a note saying the data was not used for, and it would say the reason. So now we're getting to the presentation. I'm going to walk through this report one by one, just kind of stepping through, giving information on how to read this report, and then some information to help interpret the data. I do want to mention it's complex and there's lots of factors. And so these, I won't be able to tell you exactly what is happening in your lake, but hopefully this presentation will give you some information that will help guide you. So let's first look at Secchi Disk transparency. So that's that black and white disk that's dropped into the water. When it disappears, that's our Secchi reading. That's how clear the water is. And so how do you interpret just the number? Is the higher the number, the clearer the water. The lower to the number, the more cloudy the water, the more turbid. And so what we have, this is what will be on the report. You get a summary table, and then you get a nice summary graph through time of all the data that has ever been collected for that specific lake that you're interested in. So first, I'll just point you towards the current year. So the report for that year, the current year will be on the top. It'll have the number of readings that were taken. It'll have a minimum Secchi reading and a maximum Secchi reading, and then the average. The standard deviation just helps describe the scattering of points. And then I'll talk about this more, but it gives then the Carlson TSIs, the Carlson's Trophic State Index. And that is where Secchi is used to calculate, is this lake oligotrophic, mesotrophic, or eutrophic? Now, because this is a long-term monitoring program, and because one year of data is wonderful, but more years is even better. So how do you interpret, is 15.9 average Secchi for a summer okay? Or is 11 really abnormal for a minimum? Well, we make it handy here where you can look at a 5-year window of data. So in the 5 years prior to 2015, this lake had 84 readings. And then as you can see, we can see that there was a really low sampling with a minimum at 2, but here's the average at 12. So you can start to make comparisons. So when you'd say, was 2015 a good or bad year? Well, you can still can't tell, but you can at least start to make some calls where you'd say, wow, it was a much clearer year than it was in the last 5, for example. And then also because some lakes have been involved with the CLMP since the beginning, so since 1974, there's also where it'd be all data over the 5 years. So now it's looking at comparing even longer term. And so you can see how this lake compares. It's sometimes helpful. So that's the most helpful. When you start to compare between years, that gives you a sense of what's happening in that water body. Is the clarity increasing or is the clarity declining? Or is it staying the same? Now, it is helpful, though, to compare against other lakes. And so it's handy right here on the bottom with CLMP lakes. These are all of the lakes for 2015. And so you can get a comparison of how do we fit within all of the lakes that are sampled in Michigan in the CLMP. Now, the numbers are great, but it's nice to see a general, a summary graph as well. So something that Joe showed, for some of our parameters, we have an inverted graph. Our vertical axis here is flipped. And so think of the top of the lake is happening here. And this is where you start your readings. So this is just below 10, and it's increasing as you go down. So as you can see, the trend line here is going down slightly. This is not a statistical trend, but this is a trend line just to help us get a sense of what's going on with the data through time. And what we're seeing here is a slight increase in water clarity through time. And these are the averages for that particular year that are plotted out. But you can go into the data exchange and look at your particular year between months. We don't supply that figure, but you can create that. So just to show a little bit more and to build off of what Joe showed in his presentation as a keynote, we can think of it as you're on your fishing boat at the top of the graph here. You lower your Secchi into the water so you can have a sense of the water clarity through time. So that's different from our typical graphs, which are like this, and you start reading in the bottom left. So what does Secchi tell us? Well, it can tell us a tremendous amount. It is interesting. We do ask, if somebody asks me what's happening, if they have a question about their lake, I ask, well, what's the Secchi disk? And what's the Secchi disk through time? And why that is because water clarity can indicate what's happening in the lake naturally, but then also what's happening due to human changes. It's really that pulse of your lake, that first signal that we can look at to see, is there early warning that maybe activities on the shoreline within the lake or outside the lake are having an effect on the ecosystem as itself. So, but one Secchi measurement is usually not the most helpful to know what is happening. We do want to see trends. And so looking at your Secchi through the season or annual, if you have those annual measurements, are really important, because then we can see, is your lake showing signs of eutrophication? What eutrophication means is, let's say you are classified in 1974, you were a mesotrophic lake. And let's say in 2021, you are now a eutrophic lake. So your lake has moved and increased in trophic status. And so that would be a concern that Secchi disk could be picking up. Or on another note, you can have re-oligotrophication. So that's a really big word. But really what that's saying where water clarity comes into concern is your lake was once, let's say, hypereutrophic. You have been putting in best management practices around the watershed and the shoreline. And now it's 50 years later and you're seeing the water clear up. And now you are in a mesotrophic condition, for example. And so that's a wonderful situation. And then also, I'll just bring this up and I'll talk about it a little bit more, is it also can indicate in our northern lakes, there is starting to become a concern about browning. And so, well, I'll get to it in a moment. So this is where you're getting the tea stain in your water is getting more tea stained. So I do want to just show you, so here's an example of Secchi readings through the course of a year. So not between years, just among the year. And so this can tell us a little bit about what's happening in your lake. So water clarity was the highest in May, steadily declined into July, and then started to increase again, where then we had a slight decrease in the final month there in October. And so this can start getting us clues. This could be a spring clear water phase caused by little critters in the water called zooplankton, which can explode in population in May and eat a lot of algae, creating clear water. But that doesn't last forever, and so you can start to get decline. This July could be from algae, or let's say from high recreation of boats that stir up a lot of sediments. And then as we go back, we have a slight decline here. So I'll just talk a little bit about October. Mixing occurs at that point. So if your lake is deep enough to stratify, so again, warm water sitting on top of cold water, that starts to break up in the springtime, or sorry, in the fall, when the atmosphere starts to cool down and your lake begins to mix. When your lake begins to mix, nutrients that were maybe held in the bottom parts of your lake are now up into the upper parts of the lake, where algae can bloom and you can get a reduction in water clarity. So just a note, water clarity is affected by lots of things. In combination of, it could be one thing more than the other, but they are affected by water color. You can see that here. You might be on a tea-stained lake. You could be, your water clarity could be affected by algae. It could also be by suspended solids, and suspended solids can be a lot of things like organic and mineral material. So this stream, for example, is putting in a lot of sediment into this lake, so that could impact the water clarity. Also could be from resuspension of the bottom sediments in shallow lakes. And then a neat thing that sometimes happens in our lakes, because we live in an area with a lot of hard water, is that if the temperature is right and a few other factors like types of photosynthesis that are, or the amount of photosynthesis that occurs, you can actually get calcium carbonate coming out of the solution of the water and creating little precipitate, little particles. And this is called a whiting event. And here's an example where Lake Ontario, the conditions were just right where a whiting event happened. That can also affect your water clarity. So a common question, and this goes not just for Secchi, but for all of our parameters is my reading good or bad? That is really difficult to answer. And oftentimes you have to say it depends. There's lots of factors that go into it because nothing is inherently bad. If your lake is eutrophic, that's not inherently bad. If it's declining because of our actions on the shoreline, you can consider that bad and we should be putting in best practices. So if the answer is depends, you can then ask why you might be seeing the change that you're seeing. Joe had a great example from his grandpa collecting data on Birch Lake in Minnesota, where they were taking Secchi readings. They saw that it was kind of low. They identified places that they could do better. They put in those best practices and they started to see an increase in water clarity. And that's the same in this picture as well, where in the early 70s, the water clarity was very low and highly eutrophic. They put in some best management practices in the late 70s, early 80s, and you started to see an increase in water clarity. So that would be seen as good. But what if this water clarity was because of the invasion of zebra and quagga mussels? This is where an increase in water clarity isn't inherently good because that increase increase in water clarity is due to a potential collapse of the food web. And that's being dramatic, a reduction of algae, which can have impacts up the food web. Okay, going into chlorophyll A. The quick interpretation is the larger the number, the greener the water. So what we have here is exactly the same. We have it set up where you have current year, the previous 5 years, and then all the years prior, and then comparison to CLMP lakes. A plug for later today. So here's a picture of diatoms. That's a type of algae. They're really cool, made out of silica outside. They're really neat. But here's a plug for if you are interested to learn about algae even further. There's going to be a great presentation later today by Julie talking about algae in our lakes and streams. Anyway, so you can see here where this graph is not inverted. So here's our summary graph as well, just like Secchi, but we're not inverted. So the higher the number, the more green. So we can see in this trend line that we're actually seeing a decline in chlorophyll. I should take a step back. What is chlorophyll A? Chlorophyll A is a proxy measurement for algae. So we use it to get an understanding of how much algae biomass is in your lake. So how green is your lake? We use chlorophyll A for that. So in the CLMP, we collect chlorophyll A throughout the growing season at specific times of the month to be consistent. This is because algae are really dynamic. When we say algae, there's a ton of species and they're all interacting with each other and interacting with the environment, the lake around them. And so certain algae are better at certain conditions throughout the growing season. So for example, in spring mixing, those diatoms love cold water. There's a lot of nutrients because there's lots of mixing happening there. And then there's also that movement. It keeps them aloft because they're kind of heavier algae. And so they do great in the cool months of the year, whereas they don't do as well as other algae in the warm months. Also, different things start to happen when they compete against each other in like blue-green algae, for example, also known as cyanobacteria. Some of them can take nitrogen from the atmosphere, which then makes them even better at growing than other algae. And so they grow well in warm temperatures and potentially in times when nitrogen is low because they can uptake it from the atmosphere. So this is why we collect it through the growing, through each month so that we can get a sense of what's happening in the system. Having a algae bloom, again, isn't inherently bad. Your lake might be kind of brownish, a brownish green in the springtime. That's okay. That's the diatoms blooming. That is a moment of bounty for the lake. Those algae are easily eaten by our zooplankton community. Those are the little critters in the water that eat algae. Those zooplankton are then eaten by our little fish and other invertebrates in the water, which then cascade up into the food web like fish and then into our bellies. So having algae, an algal bloom, isn't inherently bad, but it can be if there's some with that are harmful algal blooms that produce toxins. So looking at them separately is great because you can learn a lot, but also in combination can start giving us more and more clues about what is happening in that water body. So using two measurements together can help us understand. So for example, what if the Secchi transparency is low? So we have low water clarity. The question is, well, if that was it, I would ask, why is it low? Is your lake really green, or tell me more? Well, what if we also have collected chlorophyll? So now if our lake is transparency is low and chlorophyll is also low, that would then start to align to saying, okay, it might be because of if this is a tea-stained lake. And so those dissolved organic materials that enter into a lake from maybe a bog in the watershed or other things in the surrounding watershed, the surrounding geology, that can make water clarity low. And then also because water clarity is low, there's not a lot of sunlight for algae to grow. And so you actually get low chlorophyll as well. So now you can get both of them being low. Another thing it might clue you in on is that I would then ask, what's the depth of your lake? Because shallow lakes can have lots of suspended sediments, because that's when wind is blowing across, you can get wind turbulence onto the bottom of the lake, kicking up sediments. So here's just a graph showing, for example, here's how much, this is the composition of suspended particles. And the majority of the suspended particles are not algae at all, which is green. Instead, it's minerals and organic material just being kicked up from the bottom. Okay, moving into nutrients. We collect phosphorus in lakes for the CLMP. Why do we do that? Phosphorus is seen as the most limiting nutrient in Michigan's lakes, although sometimes it can be other nutrients. So you can think of this as algae and plants require a whole host of nutrients, and there's all of them are usually present in a water body. But if I added more silica, which are needed for certain algae, if I added more silica, the algae wouldn't necessarily grow because it's not limiting the growth. There's plenty of silica for that growth. But if I added phosphorus, that was the thing that's limiting how much biomass of algae is present there. And so that's what would explode. So that's what it's mean by limiting nutrient. And this is a fun experiment where they curtained off a lake. So this is one lake, they put a curtain there, they put phosphorus on one side and not on the other to see what would happen. And here we learned now about phosphorus. So we collect phosphorus in the spring and summer. They tell us different things, and they're useful for different reasons. So the spring phosphorus is collected soon after ice off. And that's because we want to collect the phosphorus, because these are both surface samples. We want to collect this surface sample when the lake is mixing. We want to do that because that gives us a sense, that gives us an estimate of all the phosphorus in your lake, because it's all mixed around. Now, fast forward to the summer months into August, September, the lake is, if you're in a deeper lake, the lake is stratified. So you have that warm water sitting on top of cold water. Now, suddenly the phosphorus that is on top of in the warm water, that surface grab sample means something different, because it's not collecting stuff. It's not collecting phosphorus that is mixed throughout the water column. And we use that measurement for calculating trophic status. And then also just a little bit more about collecting surface samples from this top during August is that if it's really high, that could indicate that there's a lot of external loading happening into your lake. And external loading means there is phosphorus coming off of the landscape into the lake. And that could be coming from multiple different places. And that can indicate that because phosphorus, when there's oxygen present, readily binds to iron and falls through the water column to the sediment. Also, phosphorus is readily used up really fast. And so algae and plants and will be will be taking that up, and everything's happening simultaneously. So you can actually get less phosphorus in the upper parts of a stratified lake. But so if you have high numbers, that could indicate to you that there might be some problems where there's a stream inlet or runoff coming from properties around the lake. Again, same setup as all of our others, where we have current year, last 5 years, and then all the years prior to that, 5 years. And we have our nice summary figures. So phosphorus is essential. It's not the lake needs it, or organisms need it for multiple different processes, but it can cause issues. So that's why we focus on it so much. And what we call, when there is a overloading of phosphorus caused by us, by humans, we call it cultural eutrophication. And so that's where we're putting in so much nutrients that you're getting a lot of algal growth and you're changing the trophic status of the lake. And that's a problem because that leads to higher algal biomass and possible harmful algal blooms. It has reduced aesthetics. Who wants to swim in this lake, for example? You get increased anoxia. So that means low to no oxygen at the bottom of the lake, which can change fisheries, as Joe mentioned earlier, and it can impact your property values. So going back to trophic status, because I've now mentioned it multiple times, but it's nice to have a little bit more to chat about, is trophic status. We have these 3 categories, but it's not categorical. It's actually a continuum. And that's my next slide. So I'll hold on to that continuum part. Trophic status, the index, the Carlson's trophic index is what we use. And that is, you take the data, you put them into although this may look pretty complicated, it's not too complicated of a formula. And then it comes out with a more consistent and comparable number that you can make comparing all 3 parameters. We average them together to get an average total or trophic state index for your lake. And we do that annually. The data is also available where you can look at them separately. So you can look at trophic state index by looking at Secchi, phosphorus, or chlorophyll separately. And that can start to tell you and paint a picture of your lake that's really helpful. But if just looking at the averages is okay to get a glimpse at what's happening. So I was mentioning that trophic status is a continuum and not just you are plunked into a category. So what the formula spit out is a number between 0 and 100, and your lake will fall somewhere in that. So think of this as like a thermometer. Oligotrophic is down here, low nutrients, high water clarity, low algal growth, low plant growth in general. And then as you go up, those things change. And the cutoffs that we use here in Michigan are at the trophic state index or the number at 38, 48, and 62. So anything between 0 and 38 is oligotrophic, 38 and 48 is mesotrophic, 48 and 62 eutrophic. And then these are the general cutoffs. So what does 38 mean to us? We can say, well, if your Secchi disk is hovering around 15 feet or more, Secchi is going to lead you into the oligotrophic classification. But now let's talk about the continuum. If you're mesotrophic, but mesotrophic right here, that tells you something more if you're mesotrophic and mesotrophic here. And so here's an example. Here's a real life example, Baseline Lake in Washtenaw, Livingston County. So they've been collecting data in the CLMP for a very long time. Here's an example of where that lake was classified in 1977. It was mesotrophic, but borderline eutrophic. Through the years, they've been putting in best practices like a sewer system, for example, but then also I believe they've been invaded by zebra mussels. But through that time period, we have seen the lake in 2002 be more in the middle of the mesotrophic, and now it's mesotrophic, but tending oligotrophic or very close. And so that's why it's important to look at this as a continuum, not just say mesotrophic, because that's not the full picture of what your data is telling you. Okay, so moving on, we have a summary panel that we give you as well, where we summarize the trophic state index for your particular lake and then compare it to all other lakes. And again, we do the current year, the last 5 years, and all years prior to those 5 years. And then we give you a nice written statement telling you about the trophic status. We also have, if you've collected dissolved oxygen, and I'll be talking about that next, dissolved oxygen would be talked about. We might also give you a recommendation. If we're starting to see things change in your lake, we might recommend collecting dissolved oxygen temperature in the following years, because it might be helpful understanding your water body. And then the general trend statement. Is the lake becoming, is it the same or is it changing? In what direction is it changing? So let's go into temperature profiles. Joe gave us a nice figure of temperature and dissolved oxygen through time when he was talking about Cisco, which is an awesome figure. This is how some of that data is derived and what you are collecting on the water, what it kind of looks like. So what we see here is a lake in the springtime, and this arrow is referring to surface water temperature. So a lake is totally mixed in the springtime in this deeper lake, so a lake that's over 15 feet deep. The lake will then start to have warm water sit on top of cold water due to the relationship between temperature and density. And we've all probably experienced this if you've swam in a lake. So if you've jumped off of your boat, you're swimming through really nice cold water or warm water, and then like really all of a sudden you hit cold water. And so that you've gone through the thermocline or the metalimnion, you're going through the zone of change. So from that warm water to that cold water below. And then once the atmosphere starts to cool off again, our lake mixes, becomes more standard temperature throughout and mixes. So what does that look like when we drop our YSI probes? So people who collect dissolved oxygen and temperature, they use a probe for the CLMP and they put it down and collect temperature and dissolved oxygen at intervals, consistent intervals down the water column. So here's an example of what springtime mixing would look like. The temperature would be pretty much the same. There could be some wiggles in there, but it'd be pretty much the same from the top of the lake to the bottom of the lake. Excuse me. And once the lake becomes stratified, you would see warm water up here because it'd be higher temperatures here. So warm water, the zone of rapid change, and then you would get usually a pretty consistent temperature in the lower parts of the lake called the hypolimnion. And then once again, once it is time for mixing, the temperature becomes the same throughout. So let's bring in dissolved oxygen into that. So dissolved oxygen is referring to my blue line. And by the way, these are for learning purposes. There's some variation in what I'm talking about in real life, but this is a good way to learn what's happening in our water bodies. So we have, when we have consistent temperature and we're mixing, we usually have consistent oxygen amounts. When the lake becomes stratified, and we're looking now at the August, September kind of side of things, we see our lake is still stratified. That's our temperature. And in a eutrophic lake, so that's the lake with lots of algal and plant biomass and growth, we have plenty of oxygen above, but then we have a rapid decline, a very rapid decline within this middle part. And then in this case that I drew here, it goes to 0 or essentially 0. And it's like that throughout. And it does this for multiple reasons. I'll just give like a quick synopsis. Is that thermocline that I mentioned, that area where like it goes from warm to cold, that acts as almost like a physical barrier for solutes within the water. And so oxygen is getting mixed up into the upper parts of the lake, but because of that gradient there, that thermocline, the oxygen is not getting down to the bottom parts of the water anymore. Now that's fine, but in eutrophic lakes, when all that algae is growing up here, it dies through the years and it settles to the bottom of the lake. Well, that dead algae, that dead plant matter, that dead organic material gets decomposed, and the decomposition process it uses oxygen. And so then in this circumstance, what I drew, there was so much bacteria decomposition that it drove the oxygen to 0 or essentially 0. And then once it mixes again, you get oxygen mixing throughout. Now, this is a eutrophic lake, but that's not all lakes. So what else do we see, again, in these idealized figures? So we could see something like this. So this is what we can see in a oligotrophic lake here. So we have our temperature again, and it's showing it's stratified. And then what we see is a lot of oxygen below, actually even more oxygen below than we see above. And that's because cold water can hold a lot of oxygen. It can hold more oxygen than warm water. And so there's plenty of holding capacity down here because it's cold. And then also there's low decomposition. And so, yes, there is decomposition, but it's so low that it's not sucking the dissolved oxygen out of the bottom parts of the water. I just put this in here because it's neat that you can see. I think if Mike Gallagher's on here, I think they see this on Gull Lake, is where you are taking your oxygen profile and you get a huge bump near that thermocline. And that bump is happening because you can actually get a bunch of algae growing on or near the thermocline, and they're sucking up the nutrients from below, but still getting enough light from above. And so they're photosynthesizing like crazy, and they're releasing a bunch of oxygen. And so you can get this bump in the oxygen in the middle parts of the lake, which is really cool. So what do we show after that quick little background of things? So on the report, we show the established stratification. This is because there's so many graphs through time, we only show one of them, but there's a whole lot more data that we show at the end of the report, but the summary just shows this. I should mention shallow lakes. Shallow lakes are really common, and those lakes typically are called polymictic. They're mixing all the time. And so they might have some times of stratification where warm is sitting on top of cold, but that can easily break up with wind or a change in temperature. And so they're mixing more frequently. Okay, so I'll just show some real life data that you can collect with dissolved oxygen temperature, which is really neat. So here is, if you look up here, this is in May, and our temperature is slightly warmer on top, but this is really not stratified yet. And our dissolved oxygen is kind of following that to some degree, but this is not stratified yet. Or if you want to consider it stratified, not strong. Now, as time goes on, this is the end of May, temperature hasn't changed too much, but oxygen has. And now June sets in, and now we definitely have a top warm layer and a cold layer down here. And we're seeing this lake still have a lot of oxygen below, but let's keep going. So here's more of June, later in June. Here's July. We're seeing that strong relationship. This is looking like an oligotrophic lake. Let's keep going. Here's August. And then in September, it mixes again. So in this lake, it was fully stratified at the beginning of August, and then boom, mixed. So what a cool, this is the data that our CLMP volunteers collect. It can tell us a whole lot. And this is just a little snippet of so what. So let's talk about no oxygen, for example. So no oxygen is anoxic. This can lead to habitat constraints. So Joe Noner talked about that with our cold water fish species. He used Cisco as an example, but there's other species like lake trout and others. And so, for example, here's a lake that is stratified and the oxygen is down to 0 in the lower parts of the lake. So you've collapsed that habitat where some fish want cold water and high oxygen. And so we probably don't see cold water fish in this lake. That doesn't mean that's good or bad. If the lake used to have it, then that could indicate something is going in the wrong direction. But some lakes just have never had cold water fish due to many factors that naturally occur for that water body. Let's talk a little bit about phosphorus release. So when you have no oxygen on the bottom, you actually get phosphorus releasing from iron, going up into the water column and becoming available for plant and animal or plant growth. Oh, okay. So that can happen. Whereas if there was oxygen down here, the phosphorus would stay bound to iron and other particles and stay in the sediment. Okay, now just talking about some more of our parameters, and we're going to talk about shorelines for a little bit. Here's a map from the Midwest Glacial Lakes Partnership showing shoreland disturbance. And what we're seeing in red is lakes that have high shoreland disturbance, and then blue going to low shoreland disturbance. This has been studied over a fair amount of time with EGLE, EPA, Midwest Glacial Lakes Partnership, all these sorts of groups. And with the EPA and EGLE working together, they've shown that shoreland habitat loss is the biggest stressor to our Michigan's lakes. We're just losing a lot of the trees. We're hardening the shorelines. We're losing all of our native vegetation, and that causes issues. This change in loss in vegetation and the increase in impervious surfaces. So like the roads, our houses are getting bigger. The vegetation is lost, or is changed to lawn grass, which is not as ideal along water edges. We've hardened the shoreline because of erosion, and this has created a major impact on our water bodies. From the loss of habitat, we have increased runoff, increased erosion, increased pollutant loading from sediment, nutrients, and a whole lot more. So it's a serious issue on our water bodies across the state. So we developed not too long ago a parameter called Score the Shore. And because if you want to begin to do something about your shoreline in your lake, you first need to collect data on where's the good parts and where are the bad parts, or more appropriately, where are the degraded locations so that maybe you can intervene, and where are the intact natural places, shorelines that you can properly protect. So Score the Shore, this is what you get in our summary report. What it does is breaks up the lakes into 1,000-foot sections, and it is analyzed between 0 and 100, 0 being fully degraded, 100 being very natural and great condition. And so then, let's see, I'll go to this one. Zooming in a little bit, what we do is we average those scores amongst all of the 1,000-foot sections to get a final score between 0 and 100. So this is 79. And then we also have it so that you can compare to other lakes. We supply other data just for interest as well as for action, is that we see where increasing density of buildings along a shoreline and the final score are closely related. And so it's important to kind of see that, where if you have more and more homes on the shoreline, you will have a lower final score, Score the Shore score. So we give a nice little summary of the best feature of the shoreline. We give problems and possible opportunities. So that's great. So we kind of help lead you towards some solutions. We also give a nice summary graph where those 1,000-foot sections, we show which ones were highly impacted, which we call 0 to 25, poor, fair, and good. And this is a map that you can create with that data, where you can see where some of your management can focus on. Where in this picture, the red was the more degraded site, where the green was the more pristine location. Going into our early detection program. So the detection of invasive plants is really important. We've developed the Exotic Aquatic Plant Watch to help collect this data. So the Plant Watch is an early detection protocol where the goal is, like this ultimate goal is to find a plant before it becomes too established, where it becomes difficult to get rid of. And so the sooner you can detect a species introduction, the better the outcome. So here's where starry stonewort is just starting off right here. You'd rather manage it at this level than here, where this is a starry stonewort meadow. And so the Plant Watch focuses on the most probable invaders. So we have a list of 5 species. We target high-risk areas for new invasions like boat ramps, public beaches. And we recommend continuous monitoring, because you may not have Eurasian milfoil this year, but you need to keep a vigilant eye, because there's always a chance that it could enter into your water body. We also have the native aquatic plant community. Plants are incredibly important. There's high diversity. It's truly like an underwater forest, as this picture kind of shows. They're valuable habitat, they help with the water clarity, and they mitigate shoreline erosion. Joe, does my PowerPoint still show, or is it saying it has stopped working?
You look great.
Okay, it may break down because it says it stopped working and I have a bad error happening. Oh, there it goes.
Yep, no more PowerPoint. Do you want me to switch over and share your slides if they're on Teams?
Let's see if it.
Looks like it's coming back maybe.
Sorry for that. We're rounding out the presentation here, so how does that look, Joe?
Looking good.
Perfect. Anyway, so there's a nice variety and diversity of aquatic plants out there, and this is a pretty intensive parameter to do, but it's very rewarding. You get to learn a lot about the different plant life that is in your lake. So you measure all species, you calculate their density and abundance, and where they are distributed in your water body. And this is important because you can start to detect how the community and species change through time. So for example, is a new species detected? That doesn't necessarily mean an invasive plant, but even just a new native plant detected. Or was a species lost through time, or multiple species? Because that could start to indicate, why are we losing certain plants? Maybe those are the sensitive species to water clarity changes, for example. And it can help you evaluate plant management. This parameter isn't made to specifically do that, but it can help give some clues to how plant management is, how well it's working. Because when you ask how well plant management is working, it's not just on the target invasive plant. You'd also like to see an increase of native species. And that would be the most successful management of our invasive species. And it might give you a sense of evaluating eutrophication. If water clarity is declining and you start to see a loss of some species and an increase in others, that could help give some indicators of what's happening. So long-term monitoring, I hope this is a blitz going through all our different parameters, but it's so important. I just want each of you to kind of think back, know a little bit about your lake and think about the history of your lake, good and bad. Maybe here's a timeline, for example, that I just quickly came up with. It's like, maybe the lake was the first cottages were put in the 1930s, and full-time residents didn't really come until the 90s. Let's say Eurasian milfoil invaded in the middle 80s, and maybe as the full-time residents came in, maybe shorelines began to get hardened, but then the lake became proactive and started to do Eurasian milfoil treatments. Maybe they installed a sewer system because there was a problem identified. And this is happening in lakes all around us, these different events, good and bad. And if you don't have the long-term data perspective, you don't necessarily see where your lake is headed or what are the impacts of that. Zebra mussels may have invaded, but maybe it wasn't big enough to have an impact on water clarity. Or maybe after that sewer system was installed, you saw dramatic change in water clarity. So having long-term data gives you it captures the history of your lake in a meaningful way that can really tell you a lot about what's happening. So thank you. Thank you all of you who are already volunteers. And if you're thinking about being a volunteer on a lake near you, I encourage you to give one of us a call, give me a call, and we can talk about that. And I'll help you through it. So thank you very much.
Thanks so much, Eric. We do have some questions that came in, and I noticed we only have 2 minutes left in our session. Eric, are you able to stay on for an extra couple of minutes to see if we can get through some of these questions?
I sure can, yes.
Awesome, great. So the first question is, Eric, can you tell us what happens to our chlorophyll samples in the laboratory?
Yeah, so the laboratory that we send them to is the Eagle, the state laboratory, and we the filter. This is the course The filters get dissolved by I think it's methanol. I can't remember what it is for Eagle. They use a certain alcohol to dissolve and extract the chlorophyll from the algal cells. They are then used spectrometry to get a sense of what how much chlorophyll is in that sample. That's really coarse, but I hope that helps.
I think that answers the question. The next one was about the trophic status index. Are the Secchi, phosphorus, and chlorophyll simply averaged for a combined TSI, or are the separate elements weighted differently?
They're not, they're averaged. But I would encourage you, if you're really interested in looking at your trophic state index for your lake, and you're really interested to see how everything's playing together, look at the averages for each parameter. Look at the trophic state index for Secchi, phosphorus, and chlorophyll, and that can start to give a really nice picture if you're interested more in that.
We also had a question come in, or someone in particular was asking whether their lake had been in the program or not. I answered that specific question in the chat because I looked it up, but I wanted to make sure that folks on the webinar here knew that if you're interested to know if some particular lake that interests you has ever been in the CLMP program, or for that matter, if a stream or river has been in the volunteer stream monitoring program, you can look that up pretty easily on the MiCorps.net website. If you go to the data exchange and do a view data search, you can look up lakes by name, by county, stream sites by watershed, and so forth. So you can easily find that information. But if you have trouble, you can, of course, reach out to us as well. So I just wanted to make a comment about that.
And it's super neat if your lake collected data in the 70s and for whatever reason had a gap of years, and you collect data. That is so helpful. Sure, it would be nice to have a continuous amount of data through that time, but wow, it's pretty neat to see what it was like 40 years ago.
Absolutely. And the lake that Carolyn asked about, we discovered hasn't been monitored since 1991. So there's a 30-year gap there. It would sure be great to get that lake back in the program and see what has changed. All right, the next program or question was about DO monitoring. Our metalimnion zone often occurs between 10 and 15 feet of depth. For several years, I took a DO temp reading at 12.5 feet, but the new website doesn't allow it to be recorded. Is there any value in knowing this information?
Well, so we don't allow it when it was in there. Joe, what did we do with
I can answer that. So right now, the website, the way it is structured for entering data is we built the data entry forms to look exactly like the paper data forms we give you. And the current paper data form for dissolved oxygen and temperature gives you specific depths at which we are asked to collect that DO and temperature data. But we are reviewing that because of situations just like yours, John, that there are opportunity, lakes have different depths. The thermoclines and such happen at different depths. And so we're considering adjusting that so that you can just enter whatever depth you happen to measure at and then put your values in there. But currently just go ahead and use the form as it's created, enter the data for the spaces that we provide. We're not losing information really about the profile. If we don't have every single measurement you collected, we still have a really good sense. And you should too, from the graphs that are created from the data that our website currently allows. All right, continue to go through a few more questions. Our lake is only 8.5 feet deep. Is measuring DO and temp even useful?
Well, yes, because having some data to show, so that's a shallow lake. That's pretty cool. 8.5 feet. Your lake is special, and it would still be interesting to know. I'll say yes, at least one time. So through a course of a year, so we can truly confirm what is happening. Shallow lakes can be kind of dynamic, and you could get some weak stratification. And that would be interesting to know.
All right, now the final question that I see in the chat is, does phosphorus bind to calcium also?
Yeah, so in a whiting event, so when calcium carbonate is coming out of solution, it will also grab onto phosphorus and settle down to the bottom. So yes, and that is a mechanism that's been identified in Michigan due to the geology of Michigan, where that's a helpful way that has been pulling phosphorus out of the water column in many of our lakes.
All right, that wraps up all the questions that we received. Thank you again, Eric, for this really great review of everything we can learn from the CLMP data. There's a lot.
There's a lot.
Absolutely. So as I mentioned before, these sessions are being recorded. In a few days, we'll have them up on the MiCorps website if you want to review them or share them with others. We also will have our Ask Us Anything session at the end of the conference at 3:30. So if you have more questions for the MiCorps team, you'll have a chance to ask them then. We are also going to be sending out an evaluation via email after the conference. So you'll have a chance to give us feedback on how you think the conference went for you. We're now moving into the lunch break. So you have an hour now for lunch on your own, and we will reconvene at 1 o'clock for our afternoon keynote. And we're excited to host Kat Kavanaugh from Ontario. She leads the Water Rangers Volunteer Monitoring Program out of Ontario, and she's going to be sharing some of her experiences with volunteer monitoring on a more international scale. So we look forward to seeing you back here then.
Perfect. And Matt, I can talk to you more about Browning. Just give me an email and we can chat.
Thanks, everyone.
Thank you.